The heart of all vertebrates beats continually, and it's actually stimulated by a cascade of calcium. The protein we're interested in studying was called cardiac troponin. So it's a heart-specific protein that binds and releases calcium, and it is central for the contractile and the relaxation response of the heart. And this is found in all vertebrate species. So when we look at land vertebrates, their cardiac troponin, this inhibitory subunit, that plays a role in the affinity of calcium binding, it has a regulatory region to it. And so when we're exercising, we put on the gas pedal and we release chemicals like norepinephrine or epinephrine, also known as noradrenaline and adrenaline, it causes our heart to beat faster. It causes our heart to beat stronger. But another thing that it does is it modifies this troponin subunit, and it's got these two serine residues that can get phosphorylated. So they get temporarily modified biochemically. And it changes their conformation. And when that happens, it removes the brakes on this troponin, the calcium binding part. And so what happens then is calcium ions can get released faster from the molecule. And this is really, really important for exercise, because during exercise, when our heart rate goes up, the amount of time that we're contracting is also going up. And what we need to defend the amount of time that the heart can fill between beats. And that's essential because the heart can only pump out as much blood as it receives. So if we shorten the amount of time that it receives blood, it's going to start pumping out less and less blood. So this inhibitory subunit, during phosphorylation, during exercise, what it does is it causes the calcium ions to be released a lot faster. And so the heart contracts faster, but it also relaxes faster. So the entire contraction response and relaxation response is shorter. This protein is extremely highly conserved in all terrestrial vertebrates, from snakes to lungfish to birds and mammals. But we were interested in looking at some of the mammals that have the highest heart rates. Because if you have a heart rate like a shrew, and shrews are tiny little carnivorous or insectivorous predators with extremely high heart rates. They truly live life in the fast lane. Even at rest, their heart rates can be 1,000 beats per minute in some species. And during exercise, their heart rates can even be about 1,500 beats per minute. As animals get smaller and smaller, their oxygen requirements actually go up. And one way you feed higher oxygen requirements is to increase your heart rate. I mean, we do this during exercise, right? It helps us deliver oxygen to our tissues faster. And for shrews, as they got really tiny, smaller and smaller, their heart rates had to continually go higher and higher and higher. And at some point, there was really not enough time for them, even at rest, for their hearts to fill properly. So effectively, what they would have to do is keep putting adrenaline all the time, which is not really an effective strategy. So what it looks like what they did, some ancestor of shrews, is they deleted this region of the troponin that regulates this relaxation time. So again, it permanently removes the brakes on heart relaxation time. And this allowed them to actually evolve even higher heart rates. So we get to the 800, 900, 1,000 beats per minute, even at rest. So we were interested to see how this evolved and such. So I started looking through some of the genomes of bats and some heart transcriptomes. I noticed that they seem to actually express two different types of troponin. So they seem to have kind of the best of both worlds. They have some proteins with it and some without it. So this troponin, this inhibitory subunit, is extremely highly conserved. And the reason why this has some relevance to human medicine is during certain types of diastolic heart disease, the heart itself, as the pump itself starts to become less and less effective, there's less time for it to fill again because it's pumping out less, so it's receiving less. And eventually, I mean, the heart tends to enlarge and initially compensates, but then it starts to dysregulate again. And the body responds to this by sending more and more signals to increase heart rate, right? To increase its pumping out. But the problem is this region of this gene starts to no longer respond to that adrenaline signal. And so these serines are no longer getting phosphorylated. And one thing that's really interesting is when we analyzed the splice sites, humans actually have an evolutionary predisposition to potentially allow it to get excised. Even though we know of no natural occurrence of this in humans, it has that innate ability to be skippable. So what we're really looking into is how bats are specifically able to insert or keep this out. And from that, we hope to apply that now to humans for the treatment of diastolic heart disease. Right? And so this would increase cardiac performance for individuals with these types of heart disease. You never know what a basic discovery will lead to, but nature, as it turns out, has almost all the solutions to so many problems, especially when you look at animals at the extremes. A little shrew or a little hummingbird with heart rates over 1,000 beats per minute, that is as extreme as it gets for a vertebrate. So if there's going to be any animals that are going to be showing these sorts of adaptations, it's going to be those ones. Much of my research over the last 20 plus years now has focused on hemoglobin, right? The molecule that binds and releases oxygen in our body, to better understand how the molecule evolved. In the last few months when we're finishing the manuscript, we were deciding where we should submit it, and the story just seemed too perfect, just too ideal to go from the smallest mammals with the highest heart rates on the planet with potential therapeutic applications to humans. And we're thinking of journals, and my collaborator William said, "What do you think about science?" And I said, "Well, you never know until you submit." Right? And so we ended up submitting there. It did take us about a year after our first submission before it got published because of additional experiments. But in the end, the paper is much stronger. It's a much better piece of work, and it's something to be proud of.